And now we will be talking about a different technique. It will be flow cytometry. I will have to explain you a little bit about the basics so that you can understand the technique. And then we will talk a little bit about some applications, okay? Also focused on genotoxicity. So we need to learn how the equipment works. And there is three main components of the equipment. That will be fluidics, optics, electronics. And then I will also talk a little bit about data analysis. In order for you to understand this, I will also have to address fluorochrome's compensation and antibodies. And then just a slide on the equipment best practices. So what is flow cytometry? So this is basically a technique that will measure and analyzes multiple physical characteristics of single particles. So it will be cells as they flow in a fluid stream through a beam of light. So as I was saying, you will have to consider the light source. It will be a laser, we will see in a minute. And then you have here the fluidics component, where you have a sample getting in, and then it's going to be directed, and the light will go through the will illuminate the particle, and then you will need detectors to detect the signal of this. So looking now at fluidics, so we will need sheet fluid to transport the particles in the stream to the laser beam. So you can see here, these are some keywords about fluidics. So you have here the sheet of fluid. You have here the sample, and the sheet of fluid will help the sample to move through this path. Depending on the sample flow rate and the sheet fluid rate, what will happen is what we call the hydrodynamic focusing. And depending on the flow rate, this focusing will be wider or narrower, and that will also have consequences on the number of cells that will pass through this beam at a time. So our main objective is that we have one cell at a time, okay? And this will be really important. You will see in a minute, especially when we analyze the DNA content. We really want to have one cell at a time going through this. And this is what we call having it in single file. Then we need to consider the optics. So as I was saying before, we need a laser. So we need a laser light source. And flow cytometries can have one laser or more lasers. You need to know which lasers do you have available. And then this will be your excitation source. And then you need to consider the emission of light. So you need to consider light scatter and fluorescence. You can measure both, okay? So first, thinking about light scatter. So when we have a light source, the equipment will be able to detect two different scatters. It will be able to detect the forward scatter because it will have a forward scatter detector in front of the laser beam, so it's parallel to the laser. And this forward scatter actually will detect the size of the cell, okay? So by passing in this angle, it will actually measure the size. Then we will have a second detector, that is a side scatter detector, that is positioned in a 90 degrees angle. So this means that by passing the light in this direction, it will provide you some information about the complexity of the cell, okay? So about the content. So this is what you can see in terms of light scatter. Of course, I will explain you a little bit better in a few slides how you will see data in the end, but it's pretty much this. You will have different types of plots. This is a dot plot, and you will see the signal of each detector. So here you have the signal of forward side scatter, and here you have the signal of side scatter. And you can see that the equipment will just create a dot for each cell, and you will have an idea about the type of cells that are going through your equipment. So you will have cells with lower size and lower complexity. You will have bigger cells still with lower complexity, and then you have bigger cells with bigger complexity here in this case. And this is actually a very common case. This will be what it's usually done to characterize blood cells. Okay, so here you will see lymphocytes, then you will see monocytes and neutrophils here. Okay. Before talking about fluorescence, I need to talk about fluorochromes. Okay, so fluorochromes are actually molecules that you can use, and they have the ability to absorb light at a certain wavelength. And when they absorb light at that wavelength, what happens is that the atoms get excited and they move from a normal energy level to a higher energy level. And very quickly, they will have to find a more stable level of energy. That is the one that you see here in S1. And there is some energy that is dissipated as heat. And then after a while, the atom will go back to the usual state of energy, and this is the energy that is emitted. Okay, so this means that for each fluorochrome you will have a spectra, an absorption and emission spectra. This is what you see here. So for this fluorochrome that is FITS, you will see that you have a maximum of excitation. So this is the light that the fluorochrome is able to absorb. Okay, and then there will be you have here the wavelengths at which the fluorochrome is able to emit. Okay, so you will have this. You need to have this into consideration when you choose the fluorochromes. We will see in a minute how we do this. So if we have this if we need to consider all this when we use fluorochromes, then our equipment, our flow cytometry, cytometer, I'm sorry, will need to have a number of filters. Okay, so we will have to include in our cytometer filters that are able to select the light that we want to detect. There are a number of different filters. These are just some examples. You have short pass filters, long pass filters, band pass filters, and then short pass mirrors. So the flow cytometry will have these filters. So in the end, what you have for the in the cytometer is has you seen the fluidics component. Okay, and then in terms of optics, you have here the excitation laser, and then you have a number of different filters that will be able to detect different colors. Okay, so you have here the ones that I told you about scatter. So this will be forward scatter and side scatter. So this will provide you information about size, about complexity, and then you will have detectors to detect fluorescence. Okay, so you'll have the FL1 for the green light, FL2 for the yellow, and FL3 for the red light. So when you work with your cytometer, when you're implementing any method, you need to consider which lasers do you have, so which molecules can you actually use to excite, and then you need to consider emission, which filters do you have, and you need also to consider another phenomenon, that is the possibility of having overlapping emissions. And this is because if we have in our methods the use of more than one fluorochrome, then we can have emissions that are very close, at very close wavelengths. So what will happen is that we have the possibility to have something like this. So this is the emission spectra of two different fluorochromes that are oftenly used together. That is FITS and PE. And you can see that if you use this the second one, the 58540 nanometers filter, you will be looking at PE, but you also have a high amount of FITs still emitting at that wavelength. Okay, so this means that you need to consider this when you implement the method, and you will have to compensate for channels. I won't get into much details, but what we can do is that we can buy commercial beads to do that, so that we can correct with the equipment which signal is actually overlapping, which signal is fits, and it's being read in the PE channel. Okay, so but we can also do this manually, just using single stained samples. Then we have the third component, that is electronics. So basically, when you start working with the flow cytometry, when I started working with flow cytometry, my first impression was, well, I can do whatever I can. I just increase the voltage and cells will be moving all around as I wish. And it's kind of strange. And this is because when you use the flow cytometer, one of the components of the electronics are actually photomultiplier tubes and an amplification system. And that means that as you increase the voltage of those amplifiers, the signal will increase. And by increasing, your population will move in the graph. Okay, so you need to consider that. You need to set up your voltage for a certain assay, and then you shouldn't move your voltage much from there, okay? So from day to day, in your experiments, you shouldn't move the voltage. You need to optimize the voltage in the beginning and then just use the same, okay? In terms of what we analyze with electronics, we call this a pulse. So what we will see is that as the particle moves and passes through what we call the interrogation point in the laser, what we will see is that the signal will increase, will reach the highest point, and then the signal will start decreasing. Okay, and this is actually what we will see later in our data. So what we can consider for these poles is that is height and area, and this will give us the information about the intensity. But we can also consider the width. The equipment will also tell you which is the width. But this is related to the time during which the pulse occurs. So this means that if you have a larger cell, or if you have two cells that are aggregated, your width will be the width of your pulse will be wider. And this is also very important when we want to distinguish the aggregates. We will see this in a minute when we talk about the application. So as I was saying, signal intensity can be measured by height or area, and the width measures the time that the cell spends in the laser. There is also another thing that I want to point out, and that is about thresholds. So there are many methods in which we don't want to see everything. Sometimes we have debris, sometimes we have autofluorescence, sometimes we have particles in our sample. And in that case, we can choose to have a threshold. And that is pretty much saying to the equipment that we just want the equipment to collect information about particles that are above a certain criteria, okay, above a certain signal. So this is what we call the threshold. So you need to be careful when you do this, because in many equipments, when you set the threshold, everything that is below that criteria will be lost. You'll never have information about that. So there are usually, at least I prefer to have it, or not to have this threshold and then just delete it delete this data at the data analysis stage. And then we need to consider software. So this is quite important. Actually, many people choose their flow cytometer based on the features of the software, because it is really important to have a good software for your flow cytometer. So the flight usually when we what we want to do, and usually what we do in any method, regardless of the purpose of the method, is that we usually start by distinguishing cell populations, and we do this by using forward scatter and side scatter, as I was showing you. So this will plot size against complexity. And with this, you will be able to see different populations in your sample. And you can use dot plots. It was what I was showing in the previous slide, or you can use also contour plots. Most of the softwares will provide you these two possibilities. Then what we do is to create regions of interest. For example, so in this case, imagine that you are interested just in lymphocytes. So you create a region around here, and softwares will allow you to create oval regions or circular regions, or even dot to dot regions. So you will have a number of possibilities to create a region, so that in the next plots that you create, the results will be focused only on this region, okay? And then there is also the possibility to have different kinds of graphs focusing only on one parameter or two parameters. So in the case of a single parameter, you will have like an histogram like this. So you will have a count on the y-axis, and you will have either any of the detectors on the x-y axis. So you will have here either forward, side scatter, or fluorescence, okay? Any of that can be here. And you will see peaks, and these will correspond to your populations. For example, if I had side scatter here, for this sample, I would have basically two populations, as you see here. So I would see a lower complexity population and a higher complexity population, okay? And then we can have two parameter plots. So you can have side scatter or forward scatter, wherever you want, you can plot it here. So this will correspond to the any of the detectors in the Y and X axis. So I'm not sure if you noticed that, but when I'm showing here the graphs, there is the possibility to use here either linear scale or log scale. Okay. So how do we choose log or linear? Well, I can tell you that, for example, for DNA, when we know that the differences are usually quite small. So we will have the usual amount of DNA or the double amount of DNA if cells are in division. So usually in those cases, we use the linear scale because we have smaller differences. But when we have very big differences, we need to use the log scale, I'm sorry. So you can see here the difference between having the log or having the linear. And as you can see, in this case, the log scale will have the ability to increase the resolution of these 6 peaks that if you have the linear scale, you will have them all very close, and you won't be able to distinguish them. So we usually use the linear only when we know that there are smaller differences. The good thing about this is that you can try everything in the beginning. So you can try all kinds of graphs, you can try all kinds of scales, you can try to use area or height or Y, and you can try to see what is best working for you and for your data interpretation. So what can we do using flow cytometry? So basically, we can analyze the expression of cell surface or intracellular molecules we can characterize and define different cell populations in heterogeneous cell population. We can assess the purity of isolated subpopulations. We can analyze cell size and volume. And for that, as I was showing you, we are looking at the relative size of the particle or the cell, the internal complexity, and the fluorescence. So this means that by knowing this, you can see that we use them a lot, and probably the most used the most common technique is actually for immunophenotyping. But you can also use it for cell cycle analysis. You can use it for apoptosis detection. And you can use it for, I didn't put it here because you will see in more detail other examples of applications that we will present now. Of course, that's to use it for all these techniques, there is another concept that I haven't talked yet, and it's about the use of antibodies. Okay. So in order to identify these populations, as I was showing you in the previous graphs, we can use forward scatter and side scatter, but sometimes that's just not enough. I mean, there are some populations, cell populations that have two similar sizes or two similar complexities, and they will still be overlapping in those graphs. So we need to identify them or distinguish them using fluorescence. And to use fluorescence, we usually use antibodies. Okay, so whenever we have available, or we can also try to do it in-house, we will need an antibody that is conjugated to a fluorochrome, okay, and that will be what will identify our cell of interest. So in this case, we are talking about cell surface markers, and we have here two examples of antibodies that can be used to detect that surface marker. So in this case, we will have a primary antibody. So you'll see an antibody that is directly detecting our molecule of interest. Or we can also use indirect antibodies. So we will have an initial antibody and then a second antibody that is the one that is that has the fluorochrome that will recognize the first one. Okay, so this has some implications in terms of sensitivity of the assay. So sometimes if we want to increase the sensitivity, we will use the indirect antibodies. But for sure, using direct will be much quicker and much more simple. So in terms of equipment best practices, so if we look into the into the different components, so if we look at fluidics, we need to make sure that it's free of air bubbles and debris, and it's pressurized the whole time. And then you also need to make sure, and this is a problem also of fluidics, it could be in the first point, it's about the clogging of the equipment. And this is something that it happens very often when you work with flow cytometry. So you need to make sure that you don't have much aggregates in your sample, because if you have them, they will clog your equipment. And you need to start everything all over again, because you will have to clean the equipment and start the sample again. So we usually use cell strainers, but this can also implicate another problem, and you will see in a minute what that problem is, and it's related with cell loss. Okay. And then regarding optics, you need to check your equipment regularly. So most of the equipments will have internal controls. So you have fluorescent beads to check if if the channels and if the detectors are working properly. And then you should use similar voltage in inter-day experiments. So as I was telling you before, this is very important to make sure that you have comparable results. So now we will go to to an example. So what can we do with flow cytometries? If we think about genotoxicity testing, let's talk today about the H2AX phosphorylation assay. So you have heard already about DNA repair, and this is an assay that is related with DNA repair. So I will tell you a little bit about the background. So what is H2AX, the kinetics, the available methods, then the method itself, the gating strategy and the data analysis, then sample and controls preparation, then a few tips for method optimization and how to plan your experiment. So what is a phosphorylated H2AX? So yesterday you heard about the DNA structures, the DNA structure, and you heard about nucleosomes. So the histone is a component of this nucleosome, and namely H2A histone, you see here. And H2AX is a variant of this histone. But it has a very specific function in DNA repair. So what happens is when you have, when you have a double strand break, the histone will get phosphorylated in a specific serine, an amino acid. So in serine 139, position 139, and that will pretty much signalize to the cell that there is a double strand break in that position, and the machinery of the cell, it gets recruited for the repair, okay, to initiate DNA repair. But of course it is important to know about these the kinetics of these biomarkers. So how do they form, for how long, and this is also very important to consider. So yesterday, when we were talking about human biomonitoring, you heard that, for example, for benzene, you have a window of 8 hours to collect the samples in order to make sure that we can actually find that metabolite, right? And here it's pretty much the same. I mean, we need to know for how long, how big is our window to actually see this phosphorylation. Because if it disappears too quickly, maybe we are just not seeing it because we didn't collect the sample at the right time. So this graph shows the H2AX phosphorylation response with time. And you can see that the accumulation, the response of H2AX is pretty quick, and it goes from minutes to a few hours. There are lots of studies about the phosphorylation of H2AX after exposure to ionizing radiation. That's pretty much most of the publications that you will see, and they will show that it goes from 30 minutes to 2 hours. So it's very quick, and after that period, the levels of phosphorylation will start to decrease because it will start with DNA, the cell will start with DNA repair. So everything is very quick, and so our window is really small. So this means that, for example, when we are analyzing populations exposed to ionizing radiations, because it is an occupational population, and because we know when they are exposed, we actually have the possibility to have some sampling pretty close to exposure. That's good. But it is also used in many other situations, and even after many days or months after exposure. But what we do know is that after this very quick phase of DNA double strand breaks repair, the cell gets into this slow phase. So the levels will still be increased at very low levels, but still increased for several months. And this is our opportunity to actually use the phosphorylation of H2AX for a long time as a possible biomarker of exposure. So there is a very nice paper that I'm just showing you here. One of the possibilities for this persistent H2AX phosphorylation is about the possibility of these kind of damage accumulate in the telomere section of the DNA. So it's quite nice. If you have the curiosity, you can take a look at it. It's one of the hypotheses. So this means that the phosphorylation of H2AX is for sure a biomarker of DNA double-strand breaks. It has been used, as I was saying, to measure, to analyze double-strand breaks induction after exposure to a number of different agents, and we can use it both in vitro and in human populations. So when we talk about observational, so population-based studies, it was used for in cancer, in populations with cancer, also in groups that were undergoing cancer therapy, also to analyze the drug resistance, for environmental and occupational screening. As I was saying, ionizing radiation is one of those examples, and aging and disease. And this is because what I showed you before. And in terms of experimental studies, so this biomarker has been used both for to study double-strand breaks response or double-strand breaks repair. In terms of matrices, you can use peripheral blood cells. This is the most usual matrix to use, but you can also use solid tissues. So the procedure is not very different of what is used in COMET assay. You just need to isolate cells. You need a single cell suspension, and then you can run the assay. And it has already been used in exfoliated cells, in buccal cells. So which techniques are available? There are a number of different techniques available. And so this one, microscopy, is actually the gold standard. So what is done is that cells are incubated with an antibody that is conjugated with a fluorochrome, and we are able to look at the microscope and count the number of foci in each cell. Okay, so this is the gold standard procedure. But there are also people using Western blot that is measuring protein levels of phosphorylated H2AX in cell lysate. And but today we will talk about the possibility of using flow cytometry. Okay, so in this case we are also looking at single cells, but we are looking at the intensity of the signal. Okay. So the advantage of using flow cytometry is that we are able to collect information from a high number of cells in a very short period of time. As you can imagine, it is much more quicker than doing this using microscopy. And at the same time, we can use multiparametric analysis. So as you have seen before, one of the possibilities of flow cytometry is to use different fluorochromes. So there are many researchers that do H2AX phosphorylation at the same time that they are analyzing cell cycle, for example, or apoptosis. There are a number of things that you can do at the same time. Among the disadvantages, it is the main disadvantage. It is that is less sensitive than the microscope. There are a number of papers trying to compare both techniques, but because you are not looking at cell one by one as you are looking in microscopy, you will lose some sensitivity. There is a possibility to have some background effect, and this is related to all to the usual disadvantages of flow cytometry, and then the fact that you need to obtain single cells. So this can be an additional step in the sample preparation. So in terms of the method, and today here, just for presentation purposes, I will talk about the method that we use in human lymphocytes. Okay. So you have different commercial kits available. So if you wish to do this in a simpler way, you just buy the commercial kit and you follow the manufacturer instructions. Or you can look at bibliography, and I have here just two examples of the papers that describe the method quite in detail. So you can try to implement the method based on these papers. So our method is based on those papers, and it has three essential steps. The first step is self-fixation and permeabilization. So because this is we want to look at the nuclear material that is in the nucleus. So we want to assess the nucleus. So we need to first to fix, so to stabilize the cells. And we also want to permeabilize these membranes. So we use formaldehyde and ethanol for that, okay? Then in the second stage, we will incubate the sample with some conjugated antibody. And in fact, we want to label both the H2AX phosphorylated and also DNA. We want to label both. So you have commercially available monoclonal antibodies. So you have here, for example, the antibody conjugated with FITs, conjugated with PE, or conjugated with Alexa fluor 488. For DNA, you have also different options. There are a number of fluorescent dyes that you can use and that will stain DNA. But as I was saying before, you need to consider when you choose any of these reagents, you need to make sure that your cytometer has the right laser and has the right filters to use it, okay? So either you look at one of these complex tables. This is just a section of a very big table that has different fluorochromes, and it has the laser that you can use, the emission, the maximum emission, okay? So this will help you to choose both the laser and the filter, or you can also use something that is available in different websites, that is a spectra analyzer. And I'm going to show you very quickly what this is. So it's something like this, where you have the possibility, and let me, for example, show you the one that we use. So we use Alexa fluor 488, okay? And you can choose to see here the excitation spectra and the emission spectrum, okay? And then you can choose here the laser. Which lasers do you have in your cytometer? And I have a 488 laser. This is the most common, the blue laser. And you can see that the excitation laser actually crosses very well the absorption, the excitation spectra. Okay, so this means that this is good for my cytometer. It has it relates with my laser. And then regarding the emission, I need to include here which filter do I have. So I have 120, something like this filter, okay? A band pass that will, something like this. Okay? What should be happening, and it's not happening, is that it should be showing you a band just here in 5, okay? Here. So it should be showing you here a band. When you write here 525, 530, it would have a band here in that wavelength, okay? And that would show you how close it is from the peak of your emission, okay? And you can do this with many different fluorochromes or fluorophores, it's the same, just to show you if you have the right laser, if you have the right filter, and how the different fluorochromes are overlapping, or if they are overlapping, okay? And if you need that information about compensation. So going back now to the presentation. Yeah. We have the final stage. So after this, we have the flow cytometer. So what we do in practice in the lab is to fix the cells for 15 minutes with formaldehyde, then to centrifuge, and then put them in ethanol overnight at 4 degrees to permeabilize them. Okay, and then in the next day, we centrifuge, we wash with PBS, we incubate with the antibody, and then we wash again to remove everything that was not recognized in the sample, and then we incubate with propydium iodide. Okay, so this is what we use to label DNA, propydium iodide. And then we analyze using the flow cytometer. We use the medium flow, and we analyze 10,000 events that are marked with PI. So this means that we will analyze 10,000 nucleated cells. In terms of gating strategy, what we do, as I was telling you before, is that we start by identifying the population. Okay, so for that we use the forward scatter and the side scatter. Okay, and this will help us to identify our population, and then we want to discriminate and exclude doublets. Okay, so remember that I was telling you that we want to make sure that we have single cells passing through the interrogation point one at a time. So there are different strategies to exclude these doublets, and these doublets are cells that are together. So something like this. This will be a single cell, this will be a doublet. So as I was saying before, what happens is that when this occurs, in the pulse, you will see that the Y will increase. Okay? So this means that in your final graph, when you plot fluorescence, when you plot Y against area, you will see cells that for the same area, they will have broader pulses. Okay, and the fact that they have broader pulses mean that you have these kinds of events. So you have cells that are passing together, and you want to exclude them. Okay. So when you want to exclude, you just create a region here, and this will be R2. Remember, here we have created a region in the population of interest. So in the next plot, you see here that the plot is gated on P1, R1. So here you can only see what is here. Everything else was excluded. And now we will do the same. We are just interested in this, the ones that have narrower pulses, and we will exclude everything else. So now in the next plots that are gated in R2, what we do is that we will plot on one side, we will plot the red fluorescence here, and here we will plot the green fluorescence. So here we are looking at PI labeling, okay, propydium iodide. So we have here the ones with no staining and the ones that are stained. So these are nucleated cells. And then we have here the labeling of Alexa fluor. So what is labeling our phosphorylated H2AX, okay? So, and this will be how it looks, a negative sample, and this is how it looks, a positive sample, okay? So you have cells that are positioned in the same place for the PI because they are nucleated cells, but then you have increased signals for the phosphorylation of H2AX. And in our lab, what we do is that we create these quadrant regions, and they will just show you the negative, 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 positive, positive, negative, and positive, positive regions for each of these channels. And then we just analyze those that are marked with PI and the fluorochrome, divided by the total number of nucleated cells, and we multiply by 100, and this will be the percentage of our positive phosphorylated cells. This is one strategy. It is not the only one, okay? So there are some labs that they will measure the median intensity. That is also a possibility, okay? So what they do is that they use an histogram. So they will plot the count against the signal of the Alexa fluor. and they will show you the median C. So another point that you need to consider is sample and control preparation. So for the samples, you need to make sure that you have single cells. So many labs use the cell strainer. That is pretty much a filter that you can move the cells through that filter and make sure that they get individualized. You need to make sure that you have enough cells. So this is quite important, because if you do it in vitro and you are using different kinds of treatments, you know that you will expect higher levels of phosphorylation. So maybe you can actually start with a lower number of cells. But when you're doing human biomonitoring and already expecting low levels of phosphorylation, what can happen is that you will have a very low number of positive events. So in that case, you may need to increase your sample population. And there are many occasions when we see that we are losing cells throughout the protocol. So make sure that you have enough cells. For human biomonitoring, there is also now a few papers showing that we can use whole blood instead of using only lymphocytes. And we saw today in a previous presentation some of the advantages of using whole blood. So you saw that to prepare lymphocytes, you would need to complete a number of steps, and to use whole blood, it will be much more simple. So there are some studies that have already tried to do that in whole blood, and it seemed to work. And then we have the fresh frozen problem, and it was also addressed today. So we have pretty much some of the same problems for all the assays. And in this case, well, what we saw in our lab is that the best thing to do is use fresh, because the freezing thawing will actually increase the basal levels of H2AX. And it's quite simple to do that when you do in vitro. You use it fresh because you choose the hours of your experiments. When we do human biomonitoring, sometimes it's not that simple. We really need to freeze the samples in some cases. So there are also some studies I can give you if you want them, that try different strategies to see what can be done after freezing, and compare it with fresh samples. And some will stimulate cells after freezing, and others will just put them overnight with cell culture medium. And this is to force cells just to leave the quiescent state that they get with the freezing. But you will always have differences. It will never be the same as analyzing it fresh, okay? But you need to, I would suggest you to make a few samples and see what are the differences, if you think that will really impact your result or not, and then make a decision. But be sure to not forget this 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 question. So in terms of controls, we will also need controls, and in flow cytometry, we not only need the assay controls that we taught today. So we will need to have a non-negative, a non-positive. This is important to make sure that your assay is running well. But you should also have unstained samples, and what we call the fluorescence -1 samples. And this is very important for us to establish the gates. So you saw before that we have those lines for the quadrant regions in certain places, and you need to make sure that you have these lines in the right place. And the only way to do that is to have you need to have an unstained sample, and that will help you positioning or identifying which is the negative. Okay, so how will this position here? And then if you have one that is negative for Alexa and positive for PI, it will help you also to position this line here. And then if you have a negative positive, it will help you to place to identify the right place for this line here. Okay, so it's very important to have all these kinds of samples. do the experiment. And then, of course, when we have the positive, it is what it is. You don't move the lines anymore. So this corresponds to your sample. So in terms of positives controls, so what we do, we have a number of options of different compounds that we can use. We heard about them yesterday, some of them. I have here some suggested the concentrations. Okay, so this is based on the literature. You need to treat them in these cases for 4 hours, in this case for 1 hour. They have different mechanisms to induce the phosphorylation of the histone. So some of them have slightly different profiles in the final plot. But in any case, you will see an increase in the phosphorylation. So it is very important to have them. So this, as I was showing before, this is how it looks, a negative. This is how it looks, a positive. So in terms of critical steps, I have already indicated a little bit some of them. So one of them is cell loss. I can tell you that in our lab, it was our first problem. So this is particularly important when you know that some papers are not giving you the centrifugation details in G, they are giving you in RPMs, and then you don't know which centrifuge they were using. And when you try to establish the protocol in your lab, you actually don't know which centrifuge force you should use because the RPMs are not comparable. So you need to be careful with that. This is a particularly tricky after permeabilization. So after putting the cells in ethanol, you will lose a higher number of cells. Check the plastic also, because there are a number of plastics to which your cells will adhere, and then you will lose the cells. So you also want to need watch the plastic, or add BSA in the washing steps. This will prevent cells to stick to the plastic and will avoid cell loss. You should also ensure self-fixation and impermeabilization, so you know that is really important. We need the stains to get in the cells, but at the same time, we need to be careful on one hand not to increase too long the period of incubation, or otherwise we will increase the autofluorescence of the sample. And you should titrate your antibody. So this is also that is pretty basic when you do this kind of assays. So I would suggest you to do something like this. You should plot the antibody amount in microliters with a stain index, and what you will see is something like this, where you have you see the line going up, and this corresponds to not staining all the positive cells. And then after a shoulder, you will see a decrease in the stain index, and this corresponds to an increase in the background. So in practice, you will need positive cells, and you will need treated cells. You will need to treat them with different amounts of antibodies, okay? And while you do this, you should keep the same time of incubation, temperature of incubation, and number of cells, okay? Be sure to keep the same. I mean, you can try to also optimize this, but you need to optimize one variable at a time. And then you will have this, something like this, okay? So you will see a curve with the maximum intensity for the negatives, for the positives, and you should do something like this, okay? So you put the fluorescence intensity of the positive minus the negative, you divide it by the SD of the negative, and this will correspond to your stain index that you will plot here, okay? And this will help you to know which is the best concentration of the antibody. And this is actually, you will say, well, the manufacturer has done some tests. They will tell me which is the best amount. Well, he has done that for his conditions, and our conditions are sometimes different. And in fact, most of the time, we can decrease a little bit the concentration, and that will also save us some money, okay? So I found this really nice scheme about what are the steps to success. I just changed a little bit the order. I think that it could be better. So you need to get prepared. So you need to understand your assay, to understand what what is your biomarker, what do you want to analyze, and then you need to know your equipment. So you can see that this is really important. At this stage, I can tell you that when you should also rely on the technicians of the brand of your cytometer. They know your cytometer the best anyone can know. So it is really helpful to have a good connection with communication with the company. They will help you a lot also in implementation of the assays. Then you need to select your antibody and your fluorochrome, and you need to consider your controls, and then you need to consider your sample. I think that we run all these steps throughout these presentations, and that you have a little bit more understanding about what is flow cytometry and what you need to consider when you implement one assay.